Hello friends, good afternoon and welcome to EduSat live lectures. Dear friends, today in immunology we will be talking about organs of immune system. To discuss this topic we have with us our subject expert Dr. Hardeep Kaur. Dr. Kaur is an assistant professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you Amrit. Dear viewers, today we will be discussing a very important topic of immunology, that is the organs of the immune system. If you recall, in our previous lecture we had worked upon the following thing. We learnt about hematopoiesis. Now we learned that hematopoiesis is the production of all types of blood cells, including the formation, development and differentiation. Now we realize that hematopoiesis takes place in bone marrow. Now is it the normal course of development? Generally hematopoiesis actually starts during the fetal development in the yolk sac. After first 2 months, almost in the beginning of the 3rd month, the process of hematopoiesis is taken up by the fetal liver and the spleen. But after a few months, let us say by the end of the 5th month or the beginning of 6th to 7th month, this entire process of hematopoiesis is taken over by the bone marrow. So in the end of the gestation, the entire hematopoiesis is done by the bone marrow, and this continues for the adult life till the end. So bone marrow is a very important organ for the production of blood cells. Now we should also recall that in the production of blood cells, there is one very important cell, that is lymphocytes. Now lymphocytes is a type of a leukocyte which is a agranular leukocyte. Now lymphocytes constitute about 20 to 40% of the blood cells, body's white blood cells, and almost 99% of the cells that are present in lymph are lymphocytes. They circulate in the blood and the lymph and are capable of migrating into the tissue spaces and to the lymphoid organs. Now these are very important cells, because these are the cells that have the properties of adaptive immune system, that is the diversity, specificity, memory and non-self recognition, self non-self recognition. These are the 4 factors which make them the very important cells of the adaptive immune system. Now after understanding this was what we had covered in the last lecture. So we know what is hematopoiesis, we know what are lymphocytes. Now let us come to the lymphatic system. What exactly is the lymphatic system? Now to understand a lymphatic system, we must understand that it is a subsystem of a circulatory system. It is a vascular system, just like we have a blood vascular system which forms one of the most important part of the blood circulatory system. Another circulatory system is the lymphatic system, which is going parallel to the blood vascular system in the vertebrate body. It consists of complex network of vessels, tissues and organs. Now the lymphatic system helps in maintaining the fluid balance in the body by collecting the excess fluid and the particulate matter from the tissues and depositing them in the bloodstream. It also helps in defending the body against the infection by supplying disease fighting cells or the lymphocytes. Now let us further understand what do you mean by lymphatic system. Now what is lymph? Lymph is the fluid that is circulating in various channels of the lymphatic system. Now lymph is actually formed when the interstitial fluid. What is an interstitial fluid? As you can see in the figure, the fluid or the blood which is going through the blood vessel, it actually percolates out or it oozes out through the capillary capillary membrane. The capillary membrane is permeable. Now the fluid or the blood minus the blood cells comes out through the capillary membrane. It goes inside the tissue spaces around these capillaries, and it is now called the interstitial fluid. This is a fluid which lies in the interstices of all body tissues. Now this fluid which is present in the tissue surrounding the blood vessels, it is then collected through the lymph capillaries and it is then transported through these lymph vessels to lymph nodes before ultimately they are emptied out through into the left subclavian vein, where it is finally mixed back into the blood. Technically, whatever fluid which is lost from the blood and has entered the tissue spaces, some of it goes back. Now, the rest of it, which is now called lymph because it is minus the blood cells, will be taken up by the lymphatic vessels, and these lymphatic vessels will bring it back to the blood, so that the normal constituency of the blood, the normal fluid composition of the blood remains constant, it does not change as much. Otherwise, what will happen? The blood will start becoming more thicker and more concentrated if the fluid oozes out. Now, the word lymph is derived from the name of a Roman deity of freshwater, Lympha. Now, this is the root of lymphatic system. Here, as you can see, the lymph or the fluid which has come out through the blood capillaries is carried up by the lymphatic capillaries. It then goes to the collecting vessels. It then courses through many lymph nodes. Now, while moving from lymphatic capillaries to the collecting vessels, during their way, the many lymph nodes may come into play, like the fluid from the blood is collected through the capillaries. Now, before it is going into the collecting vessels, it passes through the lymphatic nodes. Now, as it is passing through these lymph nodes, it is getting richer and richer with the lymphocytes. Once it is collected through the collecting vessels, then it goes to the higher trunks, lymphatic trunks. Now, and these trunks are the vessels which are draining most of the body parts. Now, once these trunks, they join together to a collecting duct. Now, two very important lymphatic collecting ducts are, one, the right lymphatic duct, and the second is the thoracic duct. This thoracic duct happens to be the largest lymphatic vessels which finally drains into the left subclavian. So, we know the lymph is flowing through all this system of vessels, and finally this entire lymph is being finally given back into the blood through the subclavian vessel, and hence the normal fluid composition of the blood remains constant. Now, this is again similar thing which is showing that lymphatic vessels are starting with lymphatic capillaries. Now, you can see in the lower part of this, the lymphatic capillaries are the minute capillaries which are going parallel to the blood capillaries. Whatever fluid is coming out through the blood capillaries through its membrane, it is now going into the lymphatic capillary. These lymphatic capillaries are joining to form lymphatic collecting vessels, which become bigger vessel and form lymphatic trunks. The lymphatic trunks finally leads to lymphatic ducts, and as you can see, the lymphatic duct is finally draining into the subclavian or the blood vessel, and hence going back into the heart. So, the entire blood composition is maintained through the lymphatic system. Now, while the lymph is moving through this collection of vessels into the lymphatic system, what is happening? It is becoming richer and richer with lymphocytes, and we all know from our previous experience that lymphocytes are very important cells of our immune system. What are lymphocytes? If we recollect, we have the NK cells, we have the B cells, that is B lymphocytes, the T lymphocytes, and of course along with the APCs, they form the entire repertoire of cells which are very important in the immune system. Now, these lymphocytes rich lymph is passing through all tissues, or it is bathing all tissues of our body, and while bathing all tissues of our body, if an antigen comes and at any point in any tissue of our body, it will be taken up by these lymphatic vessels and carried to the lymphoid organs, where this antigen will be taken care of by the lymphocytes, and hence our body is hence protected. Now, how does lymph actually flows? We have understood the root of the lymph flow. Now, let us understand the lymph flows at a very low pressure and speed. Now, as we know in the blood vascular system, it is the heart which is pumping the blood. It is giving a pressure to the blood. We do not have any lymphatic hearts. We rather in vertebrates, what happens? The lymph is flowing at a very low pressure and speed. Now, this flow of lymph is further helped by the rhythmic contraction of lymphatic vessels and by the skeletal muscle pump, or rather by the muscles contraction, by the movement of the body, by all muscles that contract, the lymph actually flows through these lymphatic vessels. Once flowing through these lymphatic vessels, it does not have a high speed unlike the blood or a high pressure, it is a very low pressure and speed. Now, one thing which prevents the backflow of lymph in these vessels is the presence of valves in these capillaries. The valves do not allow the lymph to move back. Now, rapidly flowing blood in the subclavian is another factor which pulls the lymph it creates a pressure which pulls the lymph towards inside. So, what happens is the lymph is flowing at its own slow speed in the lymphatic capillaries, aided by the muscle movement of our body. The backflow is prevented due to the presence of the valves. The third important factor is the pressure created by the subclavian, the blood present in the subclavian vein which is moving at a very fast rate, which now pulls lymph to flow into it. You can see in the diagram there is a lymphatic vessel, the lymph is flowing in the lymphatic vessels, and as it is passing through the tissue into the tissue fluid, the high pressure of the blood is actually causing the lymph to move inside it. This is what happens in the subclavian vein which pulls the lymph to flow inside it. Now, lymphedema is a common term which is used for all blockages which occurs in the flow of lymph. Now, blockages in the lymphatic capillaries could be due to some swelling in the tissues, or it could be due to some tumor pressure, some tumor has developed and is impinging on these capillaries and causing the lymph not to flow, so leading to a blockage in these capillaries. It could be due to a parasite. As you can see in the figure, this person is suffering from elephantitis and it has led to parasitic blockage of the lymphatic channels in his leg. So, the leg has swollen so much, this is called elephantitis. Now, lymphedema could also be due to some surgery and leading to inflammation in that region in those lymphatic capillaries, hence preventing the flow of the lymph. All these reasons which are leading to the blockage in the flow of lymph are together called lymphedema or edema or swelling because of the blockage in the lymphatic channels. One example as it is shown is elephantitis. Now, what exactly are the lymphatic cells? Now, in our previous lecture, we have understood that we have 4 very important cells, the natural killer cells, the T lymphocytes, the B lymphocytes, the antigen presenting cells. The antigen presenting cells include some macrophages, from which are the macrophages are the tissue borne monocytes, or rather macrophages are the tissue borne phagocytes, while monocytes are the blood borne phagocytes. We have the dendritic cells which are present in epidermis, mucous membrane as well as in the lymphatic organs. Now, each and every cell of the lymphatic system have a relevant function. For example, in our previous class we discussed the natural killer cells. They have not the normal or the regular lymphocytes. They are the cells which are having different kind of receptors as from the T cells or the B cells. Now, natural killer cells, if you recall, have the KIR and the CAR receptor. The KIR receptors are the killing inhibitory receptors, while the killer activating receptors are the CAR receptors. And if there is an antigen present in our body cell, it is the CAR receptors get activated in the NK cells and it finally kills these antigen containing self cells. Now, the T lymphocytes, if you recall, they are of 2 types, the T helper cells, the T cytotoxic cells. Now, the T helper cells are those cells which have CD4 receptor, while the T cytotoxic cells are having a CD8 receptor. This is just to recall from our previous lecture what we did about the cells of the lymphatic system. The T cells, the T helper cells require help from the B cells, and T cytotoxic cells, instead once it comes in contact with the self cell which is having maybe an antigen in the form of virus or a cell which has gone haywire like a transformed cell, the the T cytotoxic cell will convert into cytotoxic T lymphocyte, that is CTL, and will finally devote the cell which is having an antigen. The antigen presenting cells, whether it is the macrophage or the dendritic cells, they have a very important role to play, that they pick up the antigen from outside and process it and present these antigens to T helper cells. Now, the antigen presentation takes place along with MHC1 or MHC2 depending on what cells it is presenting to. Generally, APC is presented to T helper cell with the help of MHC2 molecule. So, this is just to recall the cells which are playing a very important role in the lymphatic system. All these cells, the details of these cells can be found in our previous lecture. Now, these are the cells. Now, let us understand what is a lymphatic tissue. Now, lymphatic tissue is actually of two types, the diffuse lymphatic tissue or a very organized lymphatic tissue. Now, if the lymphocytes are present in mucous membrane, especially in case of lungs and lamina propria of intestinal walls, they tend to be present in a diffuse form. They do not have a capsule surrounding it to form it a organized state. They are very diffused in form, and this diffused lymphoid tissue is very important because it is almost covering the entire mucous membranes present in whether it is in the genitourinary tract, whether it is in the digestive tract or in the respiratory tract. Similarly, the diffuse lymphatic tissue may also compose MALT, which is the mucosa associated lymphoid tissue, which is present in passages open to the exterior. As I was just telling you, we have various avenues or various places from where the pathogen or an antigen can come in. The antigen can come in through the food we are eating. We do not know, it might be having some bacteria or a virus. So, the pathogen can enter through the mucosal membrane lining of the digestive tract, or the pathogen or a microbe can come through the air we are inhaling. So, this air can contain the pathogen, and through the mucosal membrane, maybe through the nasal mucosal membrane or through the bronchi, the mucosal membrane, the pathogen can enter and hence cause very bad situation inside, or through the genitourinary tract, like for example in a UTI infection, urinary tract infection, the pathogen is entering through the genitourinary tract. It is entering through the mucosal membrane of the genitourinary tract. So, whether it is through the intestinal, digestive, bronchi, nasal or through genitourinary tract, the pathogen is entering the mucosal membrane. Now, who is protecting? Generally, the diffuse lymphoid tissue is protecting us from all these pathogens. Now, when this the entire masses of lymphocyte is covered by a capsule, it forms an organized lymphoid tissue. So, we can define the organized lymphoid tissue as dense oval masses of lymphocytes which flock together in response to a pathogens, hence forming lymphoid follicles. Now, a very good example is a Pear's patch, where there is a more permanent congregation or clusters found at the junction of small to large intestine. Generally, it is at the end of the small intestine in the terminal region of the ileum that Pear's patches are present. If you carefully see Pear's patches, which will come in the later part of our lecture, the Pear's patches have a capsular or rather they are very dense oval masses of lymphoid tissue. So, such tissues which are very dense aggregation of lymphocytes, they are generally called organized lymphoid tissues. And with this we come to the conclusion of the lymphatic tissue, and then we go to the lymphoid follicles or further details about the lymphatic tissue. Now, lymphatic tissue can have lymphoid follicles which is consisting of an aggregates of lymphocytes and non-lymphocyte cells surrounded by a network of draining lymphatic capillaries. Now, as we have understood while defining lymphatic tissue, that lymphatic tissue could be diffused or it could be organized. So, technically when we are talking about the organized lymphatic tissue, it is an aggregation of lymphoid as well as not lymphoid cells. Now, generally most of these organized lymphoid tissues are surrounded by a network of draining lymphatic capillaries. Now, this aggregation can be in a follicular manner, it can be called a primary follicle or it can be called a secondary follicle. Now, if you can see, go back to the slide, we will see that in the slide we are showing a secondary follicle. Now, till there is no antigenic challenge, the lymphoid follicle would almost always be present as a primary follicle, which is comprising of a network of follicular dendritic cells, that is FTCs, and mostly resting or B cells, the naive B cells. Now, what happens whenever there is an antigenic challenge? A primary follicle converts into a secondary follicle. Now, secondary follicle is very different from a primary follicle. Primary follicle has an aggregation of resting B cells and FDCs. However, a secondary follicle will have two regions. One, the cortical region, as you can see in this slide, there is a very dense cortical region. If you can see, there is M written in that region, that is the mental or the dense mental, while there is a central region where GC is written, that is called the germinal center. Now, what happens after an antigenic challenge? A primary follicle gets into or changes into a larger secondary follicle. And what is a secondary follicle? It is a ring of concentrically packed B lymphocytes surrounding a center which is called a germinal center. The germinal center actually consists of proliferating B lymphocytes and an area that contains non-dividing B cells and some helper T cells along with macrophages and FTCs. Now, this is the germinal center where most of the selection of lymphocytes is taking place. Whether it is a selection of T lymphocytes or B lymphocytes, the germinal center is the region which is most important in recognizing a self-reactive or a non-self-reacting lymphocytes. If a lymphocyte is not reacting to an antigen, it is of no use to us. So, such lymphocyte would be eliminated. However, if a lymphocyte is reacting to your own body peptides or cellular peptides, again it is of no use to us. So, a rigorous selection pressure is taking place in the germinal center of a secondary follicle. Now, with this we come to the lymphoid organs. The lymphoid organs can be defined as primary lymphoid organs, the secondary lymphoid organs and tertiary lymphoid organs. Now, what happens? We have understood that in hematopoiesis that most of the cells are formed. Similarly, during hematopoiesis, once lymphocytes are formed, they enter the primary lymphoid organs. Now, what are primary lymphoid organs? Bone marrow. Bone marrow is a primary lymphoid organ and we know hematopoiesis is taking place in the bone marrow. Now, what happens? Some of these lymphocyte progenitor cells move to thymus. Now, those lymphoid progenitor cells that move to thymus will now convert themselves into thymocytes. So, technically the certain progenitor cells which are, I am talking about the lymphoid progenitor cells which remain in the bone marrow will be destined to form the B cells or B lymphocytes. However, those progenitor cells which now have moved into thymus will now convert it into thymocytes or finally form the T lymphocytes. So, technically the primary lymphoid organs are the organs which are the site of maturation of B lymphocytes or T lymphocytes. Let us go back to the slide. Now, in the slide we said that we have three types of primary lymphoid organs, secondary lymphoid organs and the tertiary lymphoid organs. We can also say that immature lymphocytes, they are generated in hematopoiesis. Now, these immature lymphocytes, once they move into their primary lymphoid organs, they are till this time they are naive, they are called naive lymphocytes. Now, they convert themselves into bone marrow B lymphocytes if they are present in bone marrow or T lymphocytes if they are present in the thymus. Now, to become immunocompetent, they need to have an antigenic challenge. This antigenic challenge is provided to them and then these naive lymphocytes can convert it into immunocompetent lymphocytes. This all takes place in the primary lymphoid organs. Then comes the secondary lymphoid organ. In secondary lymphoid organs, which are like spleen, lymph nodes, the mucosal associated lymphoid tissues, what happens? It is the place where antigen is trapped. Now, we can make it much easier by saying that the soldiers, if lymphocytes we consider them as the soldiers of the immune system, they are formed, they become competent in the primary lymphoid organs, but the actual battle commences in the secondary lymphoid organ, where they come in contact with the antigen in the presence of APCs, and lot of lymphocyte proliferation takes place in the secondary lymphoid organ. More and more of lymphocytes are produced, more specific lymphocytes are produced, the products of lymphocytes, specific products of lymphocytes are produced, which is capable of killing the antigen. So, this actual battle with the antigen takes place in the secondary lymphoid organs. Now, the entire nexus or the relationship between the primary lymphoid organs and the secondary lymphoid organs make sure that most of the antigens are killed before they bring any harm to the body. In certain cases, we also consider a specified lymphoid organ called the tertiary lymphoid organs. Now, tertiary lymphoid organs is generally a very good example is scald or cutaneous associated lymphoid tissue. Now, most of these organs are those where the lymphoid cell is imported as and when required. For example, whenever there is an inflammatory response, the preformed lymphoid cells would be brought over there and they will be finally leading to killing of the antigen. So, students, with this I come to the end of my lecture, where we have understood what is the lymphatic system, what are the cells which are very important and form the important constituent of the lymph, and these cells how they are organized, whether it is in a diffuse manner or in an organized manner to form a lymphoid tissue, and finally these tissues forming the three kinds of organs, the primary lymphoid organs, the secondary lymphoid organs or the tertiary lymphoid organs. We will continue with these lymphoid organs in our next lecture. Thank you very much. Welcome back, viewers. In this particular lecture, we will be continuing with the organs of the immune system. In our previous lecture, we have understood the cells of the immune system, the lymphatic system, the cells of the immune system, the tissues of the immune system and the lymphoid organs, which are the organs of the immune system. Let us go back to the same slide which was describing the lymphoid organs. We said lymphoid organs are of three types. The primary lymphoid organs, we discussed that bone marrow and thymus are the primary lymphoid organs. They are the actual site of maturation for B lymphocytes and T lymphocytes. Now, we understood that during hematopoiesis, the entire blood cells including lymphocytes are generated in the bone marrow by the process of hematopoiesis. Now, whether it gets into becomes a lymphocyte and which type of lymphocyte will depend upon whether this progenitor cell will stay in the bone marrow or will move to thymus. Now, if it stays in the bone marrow, it will lead to the production of B lymphocytes. However, if this lymphocyte goes to the progenitor lymphocyte cell goes into thymus, it will convert into thymocyte. Thymocyte will eventually evolve into T lymphocytes. Now, hence we can say the immature or the naive lymphocytes undergo maturation in this primary lymphoid organ to become immunocompetent. Now, once they have undergone this process of maturation, they are able to take care or they are able to mount an immune response. Hence, they have become immunocompetent. Now, there are secondary lymphoid organs where, as I told you in my previous lecture, it is the place where the antigen is trapped and killed. So, spleen, lymph nodes, malt is a mucosal associated lymphoid tissue, and malt is also again of various types. If it is in the bronchi region, we call it as balt. If it is in the nasal region, we call it as the nalt. If it is in a gut, we call it as galt. So, we will come one by one to each of these organs. So, spleen, lymph nodes, malt, these are the places which trap antigen along with the antigen presenting cells. A lot of lymphocyte proliferation takes place. So, we have enough lymphocytes and a lot of selection also takes place in these organs, where the specific lymphocyte or the products of the specific lymphocyte, specific products of the lymphocytes are produced, which will have a which will kill the antigen, devour the antigen. Finally, we said that we have a tertiary lymphoid tissue. The example we gave was scald or the cutaneous associated lymphoid tissue, which are important because they import the preformed lymphoid cells to a site, maybe an inflammatory site, site of inflammation, and hence kills the antigen there and then itself. Now, with this we come to this, if you can see in this slide, this is the human lymphoid system. The primary organs are bone marrow and thymus. Here only one bone is shown, we have plenty bones, we know that. This is one long bone, and you can see we have shown the bone marrow with the red inside. The thymus is present as a small bilobed structure above the heart. Now, the size of thymus also varies with the age. Now, the thymus and the bone marrow is a part of the primary lymphoid system. Now, you can see there is the spleen, the Peyer's patches, the appendix, tonsils, lymph nodes. They are generally all these are shown as blue, and they are all secondary organs and tissues. Now, all these together along with the lymphatic capillaries form the lymphatic system of our body. So, our lymphatic system has lymphatic capillaries and all these lymphoid organs. Together they are almost going into every tissue of a body. So, technically lymph which flows through these organs and through these capillaries is bathing every tissue of a body and is taking care that any antigen which comes to any tissue will be taken care of by the lymph present there. Now, let us go back to the primary lymphoid organ. The bone marrow is the primary lymphoid organ. It is the flexible tissue in the interior of bones. In humans, blood cells are produced by the course of the bone marrow in the head of the long bones in a process called hematopoiesis. Now, we had already discussed that in human and mice, bone marrow is the predominant site of B cell origin and development. Is it true for all organisms? No. We are just talking about the primates and the rodents, that bone marrow is the site of B cell origin and development. However, in lower vertebrates, for example in birds, a lymphoid organ called bursa of Fabricus, which is the lymphoid tissue which is associated with the gut, is the primary site of B cell maturation. As you can see in the figure, this hen is having the cloacal region is having an extension which is called a bursa of Fabricus. It is this region where actually the B lymphocytes are produced. This is the primary site of B cell maturation in the birds. Now, in cattle and sheep, the primary lymphoid tissue which is hosting the maturation, proliferation and diversification of B cells early in the gestation is the fetal spleen. However, later in gestation, this function is assumed by a patch of tissue which is embedded in the wall of the intestine, which is called the ileal Pear's patch, which is almost analogous to the bursa of Fabricus in the bird, but it is a different tissue which is present in the intestinal wall, which we call as ileal Pear's patches in cattle and sheep, and it contains a large number of B cells. This is the main site of production and B cell maturation in case of cattle and sheep. Now, coming back to the primary lymphoid organ, the other important primary lymphoid organ is thymus. As we discussed, that thymus is a bilobed organ which is present on top of the heart, just posterior to the sternum. Now, it reaches the maximum size during puberty. Now, in some cases it is said that it can reach the maximum size of almost 70 grams in infants. In adults it is only 3 grams. So, technically it starts from almost 3 grams in the fetus, moves off to, it increases in size, can go up to, generally it is up to 35 grams to 40 grams, but it can go up to 70 grams in the infants. Now, once the child has reached puberty, the atrophy of the thymus gland starts taking place. So, generally thymus is fully developed in an infant. On reaching puberty, the atrophy starts and now the size of the thymus decreases, and in old people the thymus has come back to 3 grams. So, technically the maximum size is in the infant stage. Now, 95 to 99% of T cells die in thymus. As I was telling you, in thymus, within the thymus there is a follicle present. Now, in the thymic follicle what happens, the central region is having a lot of elimination, T lymphocyte elimination. All those T lymphocytes which are actually killing or which are affecting your own body peptides, cellular peptides, they are of no use to us. Those T lymphocytes will be eliminated. Similarly, all those T lymphocytes which are not acting against the antigen, which are not recognizing an antigenic peptide, it is also of no use. So, such T lymphocytes would also be eliminated. So, technically almost 95% of the T lymphocytes are eliminated. Only maybe a subset of T lymphocytes that have formed are actually capable of mounting an immune response. Now, the thymus consists of a cortex and a medulla. The development of T cells or T lymphocytes from hematopoietic progenitor cells and their maturation exclusively takes place in thymus. Now, if you can see, the thymus in the left side is actually of an infant, while on the right side the thymus has gone down into just two flappy bilobed structure. This is an adult thymus. So, you can see how big is the thymus in the infant and how small is the thymus in the adult. Now, going into the anatomy or the histology of thymus, you can see the thymus on regular staining looks like that. We have thymus which is encapsulated by a capsule. This capsule gives in trabeculae. There is a definite cortical region and a medullary region. The cortical region has dividing thymocytes. Now, thymocytes, once they have undergone proliferation, goes into the medullary region. The medullary region is having interdigitating dendritic cells along with blood vessels, the macrophages, and of course, the T lymphocytes. This is the place where maximum elimination of T lymphocytes is taking place. This is the place where almost 90 to 95% of T lymphocytes are removed. Now, how important is thymus? This can be understood if we have a thymectomy done. If we have a thymectomy done in a neonate, let us say, in a thymectomy done in a neonate would lead to a deficiency of a cell mediated immunity in this child. Now, he will be having a recurrent recurrent antigenic attack. Now, this is further validated by the loss of thymus at an early stage through genetic mutation, for example, in DiGeorge syndrome, which results in severe immunodeficiency and high susceptibility of an individual to infection. Thymectomy, as I just told you, it is a surgical removal of thymus. The usual reason generally why do we do thymectomy, it is generally most of the time in a neonatal stage this thymectomy is done, because thymus is so big that is it is engulfing or covering the entire heart. Now, unless to access the heart, some part of the thymus or generally thymus is totally removed. This process is called thymectomy. Now, by performing thymectomy in neonate, although we are accessing the heart, but we are later this individual will have to be very careful, because most of the cell mediated immunity because of T lymphocytes he would not be having that. So, thymectomy further emphasizes also in certain nude mice also thymectomy results in high susceptibility of these individuals to infections. Now, another very important secondary lymphoid organ is spleen. Spleen is present in the upper far left portion of the abdomen to the left of the stomach. Now, this, as you can see, the spleen is a fist shaped, it is purple in color and about 4 inches long. Now, it is mostly important in blood production in the fetal stages, as we had discussed before, that during gestation most of the blood formation takes place in the spleen, while in the adults it is acting as a filter for the blood as part of the immune system. Now, blood carrying antigens as well as worn out cells will be brought to the spleen through the blood vascular system. Spleen is a region where all these worn out cells will be broken up. It is also a blood reservoir for all old red blood cells which are recycled in the spleen. Most of the hemoglobin is taken up, it is not given off, and platelets and white blood cells are also stored here. It is also helpful in fighting certain kinds of bacteria, for example, the bacteria causing pneumonia or meningitis. Now, splenectomy is been found to cause bacteremia or blood-borne bacterial infections. Or if we have splenectomy, such individuals would be highly susceptible. For example, in kids, in infants, the removal of spleen can lead to bacterial sepsis also, while in adults, bacteremia or blood-borne bacterial infection is more rampant. Now, spleen is generally not supplied with lymphatic vessels. Instead, spleen have a splenic artery. Now, antigens and lymphocytes are carried to the spleen through the splenic artery. Now, going into the histology of spleen, spleen as you can see is present just next to the stomach under the rib cage. Now, spleen if histologically will have two regions, a red pulp region and the white pulp region. As you can see, there is a capsule which is encompassing the spleen. Capsule is throwing itself into trabeculae inside the spleen, which is compartmentalizing the spleen. Now, the spleen has a red pulp and a white pulp. Now, the region which is whitish in nature is more having lymphocytes, macrophages, and it surrounds small branches of the splenic arteries. These small branches are called pulse. That is periarterioid lymphoid sheath. Now, pulse is bringing the antigen inside. It is going into the space which is filled of lymphocytes and macrophages around this pulse region in the white pulp. Now, the red pulp region is actually called red because it is having erythrocyte, more of erythrocytes. Now, erythrocytes gives the red color to this red pulp region. The macrophages and erythrocytes are more present in the red pulp, while lymphocytes are more present in the white pulp, and it is this white pulp region where the antigen is brought through the splenic branch of the splenic artery and is taken care of by the lymphocytes. Now, we also have lymph nodes which are present as secondary lymphoid organs. These are very small bean shaped glands which are present throughout the body. Now, they can be singly present or they can be present in the form of chains, one after another, in a beaded form, you know. So, they can be 2 mm to 10 mm depending, and it has been found if you see, if you dissect a mice or a rat, you will see a mice which has been bred in the lab, which is some, for example, like albino mice which have bred in the lab. They have very small lymph nodes, while a black rat found in the drains would be having very big lymph nodes. This is because the lymph node has highly developed due to the presence of so many antigenic challenge during its life. Now, in a human, the spleen size, the lymph node size is generally 2 mm to 10 mm. It is the only organ that filter lymph exclusively. Now, if you go to the histology of the lymph node, we will see that it has very few efferent vessels. So, what happens? We have many efferent vessels which are bringing the lymph, but only one or few efferent vessels. So, lymph comes through the efferent vessels and goes out through the efferent vessels. We will come to it once we will be understanding the histology of lymph node. The lymph node diseases are generally called lymphadenopathy. Now, there is a specific term which is called lymphadenitis, where there is a swelling of lymph node. Now, the lymph node could be painful or swollen up because of the presence of the foreign antigen. For example, as you can see in the figure, there are cervical lymph nodes present next to the neck. We have certain axillary lymph nodes in the armpits, the inguinal lymph nodes in the groin region. We have certain mesenteric lymph nodes present in the intestine. So, lymph nodes are present mostly singly or also in the chains in throughout the body. They are covering the entire body, and it is generally been found that lymphadenitis can take place maybe because of certain tumorous situation. For example, in most of the breast cancer, what happens, the lymph node becomes very swollen, and if the infection has or the tumoral cells have gone into the axillary lymph nodes, generally the person loses not only the breast but also the axillary lymph nodes surrounding it. This is done to ensure that the metastatic tumor does not go into the other places of the body. Now, here we are showing the histology of the lymph node. As you can see, the lymph node has a capsule, the pericapsular space. This is followed by the cortex, the yellow cortex region, the red paracortical region, and this is followed by the green medullary region. Of course, this color differentiation is done only to let you understand that these are the very important regions of the lymph node, the cortex, the paracortex, and the medulla. The capsule gives off trabeculae. As you can see, the black trabeculae present in the histological slide of the lymph node. It divides the node into various compartments which contain stroma and parenchyma, which is further subdivided into cortex and the medullary region. Now, this cortical region is mostly composed of follicles. Now, follicles can be the primary follicle or a secondary follicle. Now, primary follicle, as we had discussed before, it is generally when there is no antigenic challenge present. This primary follicle can convert itself into a secondary follicle depending upon the presence or absence of an antigen. If an antigenic challenge is there, the primary follicle will convert into a secondary follicle. Generally, the cortical region follicles are having B cells and follicular dendritic cells. Now, the paracortical region is thymus dependent. It has T cells with interdigitating dendritic cells. Now, we have multiple efferent lymphatics. This is followed by the cortex, which has having B cells, follicular dendritic cells, macrophages, germinal centers, and primary follicles. This is followed by the paracortical region, which has T helper cell, macrophages, and dendritic cells, followed by the medullary region, which has predominance of plasma cells producing or the B lymphocytes or the plasma cells, followed by post capillary venule and a single efferent lymphatics. Now, secondary lymphoid organs also have certain tissues called MALT, which is the mucous membrane lining the digestive, respiratory, urogenital system, which is having a combined surface area of about 400 meters square. This is the place where most of the pathogens are entering, and these areas are defended by mucosal associated lymphoid tissues or MALT. They range from loose, barely organized cluster of lymphoid cells in lamina propria of intestinal villi to very well organized tissues like organs like familial tonsils, appendix, Pear's patches, etc. They has large population of antibody producing plasma cells whose number far exceeds that of plasma cells in other lymphoid organs. Now, malt is also called galt if it is present in the gut. We say gut associated lymphoid tissue, for example, Pear's patches present in the lining of small intestine. Balt if it is bronchus associated, nalt if it is nasal associated, calt conjunctiva in the eye. O-malt it is organized mucosal associated, for example, most of the tonsils are called O-malt. Or D-malt if it is diffused mucosa associated lymphoid tissues. It is called lalt if it is larynx associated lymphoid tissue, or salt or skin associated, also called cutaneous associated or calt. Now, this is tonsils. The tonsils are again an organized lymphoid secondary lymphoid tissue which is of 3 types: palatine tonsils which are present at the posterior margin of oral cavity and is generally gets infected when we have sore throat and all. The lingual tonsil which is present in the tongue of the at the root of the tongue. Pharyngeal tonsil or adenoid which is present on the wall of pharynx. The main purpose of tonsil is to trap and destroy antigens which are entering through nasal and oral epithelial roots. This is galt or gut associated lymphoid tissue. As you can see, Pear's patches. Now, important point about Pear's patches which are present in the last part of ileum is that they have certain M cells, a sampling process which these M cells do the sampling which identifies the food material which is coming inside or the material present in the intestine, whether it is a normal food or it is a harmful microbe. This M cell has the unique ability to sample an antigen from the lumen of the small intestine and finally deliver it via transcytosis to APCs and lymphocytes on the basolateral side. As you can see, this is exactly what is happening in the M cell sampling process. The M cell is taking up an antigen, it is bringing the antigen inside, and once the antigen is brought inside, it is taken care of by the lymphocyte. Now, there is another tissue called the tertiary lymphoid tissue or the cutaneous associated lymphoid tissue. This is a subset of lymphoid system which is associated with dermis and epidermis and includes Langerhans cells and resident phagocytes. Epidermis also contains certain keratinocytes which secrete certain cytokines that help in producing an inflammatory reaction and function as APCs also, that is antigen presenting cells. Now, there is a process called lymphocyte trafficking, that is the distribution of lymphocyte is not a random process. The number and the representation of particular functional subset of lymphocytes is extremely controlled. It differs in each lymphoid organ or tissue as per the requirement. So, it is dependent on lymphocyte class. It is also dependent on the previous history or the stage of differentiation as well as on the antigenic specificity. This slide particularly shows the site of lymphocyte origin in the red bone marrow. Now, if it's an immature lymphocyte, it goes into the blood circulation, it enters the thymus and gets converted into the T lymphocytes. However, if it remains in the bone marrow, it becomes B lymphocytes. So, lymphocytes destined to become T cells migrate to thymus, while those develop immunocompetence in the bone marrow become B lymphocytes. In the second stage, after leaving thymus or bone marrow as naive immunocompetent cells, these lymphocytes seed the lymph nodes, spleen and other lymphoid tissue where antigenic challenge takes place. It is the place where the battle is taking place, the battle is commencing. It is the place where the antigenic challenge is taking place. The lymphocytes are brought to this secondary lymphoid organ and finally the antigen is being killed there. The entire battlefield is the secondary lymphoid organ. Finally, the mature antigen activated immunocompetent lymphocytes circulate continuously in the bloodstream and the lymph. We know, we have understood how lymphatic system is connected through this blood vascular system or the circulatory system. They both are forming important parts of the circulatory system, the lymph and the bloodstream, and throughout the lymphoid organs of the body. So, lymphocytes are finally these mature lymphocytes, the competent lymphocyte after taking care of the antigen are going into the blood as well as into the lymph and going to various lymphoid organs and taking care of the pathogen in various places. Now, For example, the lymphocyte population primarily responsible for humoral immunity, the B cells, they are very predominant in the spleen and gut associated Peyer's patches, while the T cells, which are primarily regulatory and cytotoxic, are present mostly in the peripheral lymph nodes and the skin, while the surface IgA bearing lymphocytes are present in the malt or the mucosal surfaces, while in non-mucosal surfaces such as peripheral lymph nodes or the skin, IgA bearing cells are rare, while the plasma cell secreting IgM and IgG are predominant. Lymphocytes can also segregate in vivo on the basis of antigenic specificity. For example, antigen specific B and T cells are disproportionately represented in lymph nodes or spleen challenged with antigen, and antigen specific plasma cells accumulate in tissue cells of specific antigenic deposition. So, this further shows that what type of antigen is going, what type of lymphocyte is going to populate an organ is dependent upon its requirement of that organ. So, this is how the lymphocyte trafficking in the body takes place. Finally, the evolution of the immune system. The vertebrate order differ greatly in the kind of lymphoid organ, tissues and cells we possess. The most primitive vertebrate, that is a jawless fish, have only galt, that is gut associated lymphoid tissue. It totally lacks B and T cells and hence cannot mount adaptive or acquired immune system. It is only having galt, while the jawed vertebrates possess a greater variety of lymphoid tissues having both B lymphocytes as well as T cells and hence display adaptive or acquired immunity. In this particular figure, you can see from the lampreys to the mouse how the immune system has developed. In a mammal, we have galt, we have thymus, we have spleen, we have bone marrow, we have lymph node, various kinds of germinal centers. All these are ensuring that most of the microbes or antigen which is coming inside the body are killed. However, in a lamprey or in a primitive fish, in a jawless fish, the only system which is present is the galt. With this, I would like to finish my lecture. Hope all the organs, cells and tissues of the immune system are clear to you. Thank you so much.
On that note, I would like to thank ma'am for this very enriching discussion, and thank you dear friends for watching our show. And on that note, I hope that you have been able to understand about the organs of immune system. On that note, thank you dear friends for watching our show. Stay tuned and keep watching. Thank you.